So I'm Dr. Suraj Singha, right? I'm your lab professor. I'm an associate professor with the biological sciences. And raise your hand if you're already in my lecture section as well. All righty, quite a good number. I'll say you hi. We'll say another hi on Tuesday, right? When we are doing the Q&A part. So and again, those who actually do lecture with me, if you see a particular point that is related, do raise that question too, right? If not, I'm going to ask you to wait until the end of the lab to raise questions regarding lecture. Okay. Um, so and so I'm going to be your lab professor. Well, some of you will meet me in the lecture as well. Most students call me Dr. S. It's easy, right? So feel free to address me that way. Um, and And in terms of my academic background, my PhD is in wildlife biology. I have a lot of experience working with animals. That's what I'm teaching. The animal portion in the lecture, but everything else in the lab. I am a wildlife biologist, but I do have experience with plants and animals equally. But in terms of the specialized part, I'm more of a animal biologist. But I have quite a bit of a background on plant sciences as well, and I love talking about plants. Now let's quickly go a round of introduction. I'm going to call out names. Monica.
Hi, I'm Monica. I'm from the Cape. My major is biology, and I took this major because I really love science, medicine, and those stuff. And I
Very
want
cool.
to help people like my mom that has like sicknesses. So yeah, biology is very important to me.
Absolutely. Very cool. I'm going by the screen, right? Jasmine.
Hi, I'm Jasmine. I'm a biology major too, and I'm from Bridgewater.
Okie dokie. Jared.
Hi, I'm Jared. I'm a biology major from Bridgewater. I'm a little bit out of the swing of things since I just took like a year and a half off since I'm just not good with online classes, but I didn't really want to keep stagnating. And I'm just in biology because when I get older, I want to do something that works with oncology. So,
Very cool. Matt.
Hi, I'm Matt. I'm a second year sophomore. I'm a bio major and I'm from Rhode Island.
All right. Cool. Um, Emily.
Hi, I'm Emily. I'm from North Attleboro. I transferred to Bridgewater from another school, and I'm going for a biology major also.
Very cool. Anik.
Hi, I'm Anik. I'm a sophomore and I'm a bio major, and I live like in Shrewsbury, close to Worcester, that area. And I went into biology because I'm not really sure which one I want to do, either something with medicine or like something in conservation. So
Yeah. And actually, I'm a conservation biologist. That is my like specialty, right? My line of work goes around, targets specifically how to protect biodiversity, how to protect wildlife, how to manage it. So very cool. Kenny.
Hi, I'm Kenny. I'm majoring in biology and I'm a freshman.
Very cool. Randolph.
Hi, I'm Randolph. I'm a senior. I'm from Randolph, and I'm majoring in biology.
Very cool. Alrighty. So let's get on to the syllabus. And while we get around the syllabus, I'm going to show you the Blackboard orientation. Okay, so I'm going to do some screen sharing real quick right now. Um, right. Are you guys seeing what I'm looking at? The Blackboard page?
I can see it, yeah.
Yeah, okay, yeah, very cool. So put it over here. Alrighty. So so your Blackboard page is actually right this one, right? And then let's wait until it loads, taking a quick hot second. And all right. So and many of you, I believe, are familiar with Blackboard, right? And how kind of the layout of the Blackboard and all of that. So the syllabus is over here, right? Getting started, that is the syllabus, right? And then if you go to course content, you find everything else, including the Zoom link. And this Zoom link is going to work for like pretty much everything, right? Office hours, meeting outside the classroom, the lab meeting, everything will be covered by that. And then we do have a self-enrollment component going on, but self-enrollment is for the common Blackboard page. It doesn't take that long to enroll there, but just to make the life easy for you, whenever there is something in the common Blackboard page that you require for the lab activity, I am going to copy those into the Blackboard, into your Blackboard that you already have access to. Okay. Here you are going to find lab manual, right? So if you have purchased it already, great, that's fine. But if you couldn't purchase it, everything you need, the entire lab manual is there, right? In PDF forms, free of charge. And evolutionary tree information, that is actually the lab we are going to do today, that's up there. And all the other labs are actually here. And I think I have arranged it in the order of appearance, right? And everything you do, all the homework assignments, you submit it through here, right? So here, it's actually the homework assignments. All the homework assignments are up here, and it actually, when you click it, it shows you the when it is due and everything as well, right? So, right, and then if you go back over here, course contents. So where do I find my homework activity? Well, so if you click this particular link, right, today's lab, you are going to find the handout, right, as well as a Word form. Like if you like taking notes on the handout, that's why I uploaded the Word form. And if you like PDFs, that is also available. So homework part is, again, if you like a PDF version, it's over here. If you want, since I believe most of you guys like to work electronically, like type in stuff. So because of that, I uploaded a Word version of the homework and a PDF version too, right? PDF version is uneditable, but the Word version is editable, right? And then of course I have uploaded the today's PowerPoint presentation, right? Over here, right? Not this one, that one, right? And then and this one is actually kind of additional information. We will try to take a look at that as we go. This is actually much more a comprehensive version that includes all the information you need. This is sort of a dumbed down version, right? And then this one contains stuff that we are going to cover today in terms of the first activity. Okay, any questions thus far regarding the Blackboard layout? Right, very good. Let's navigate on to the syllabus, and I'm trying to make sure that you can see what I'm looking at. You guys seeing what I'm looking at? The word, sorry, that's not what you should be looking at. Give me one hot second. Are you looking at the syllabus with me, or are you still looking at something else? Syllabus, yeah? All right, cool. Right, so and here, that's my name, my email address, and you already have that. So these are the office hours, right, that I have. And I kind of keep office hours very broad because, it's kind of easy to handle multiple things at the same time, right? So the office hours, I would rather frame office hours as Q&A sessions, right? Question and answer sessions. So regarding anything that we wanted to discuss, the Zoom link, right? And again, just to make the life easy, it's the same Zoom link for lecture and lab, right? For office hours during those times or outside. So it kind of makes what you want to click easy. It's just one link to get to me. And then course outcomes, you can actually read them on your own. How are we going to grade you, right? There are 9 homework activities. 2 practicals, 1 zoo, 1 botany, 2 notebook checks, okay, and then one 2 notebook checks. That's because actually before each practical, and then 1 group project, right? So the group project, I'm actually later down the road, I'm going to ask yourself to pick your own groups, right? I assume that some of you know each other, right? But if you so that's why I let you pick, your own groups, because I fairly certain that you guys know each other. Some of you don't, but that's okay, right? I will not let you, like, cut you loose or anything, but I prefer to let you pick your own groups. We will talk about that project later on. Course format, it's semi-synchronous, meaning like most of the weeks we are going to meet like this, right? And then I'm going to go over the lab activity, right? And then I'm going to at least stay around you, finish some of the more critical lab activities. And then the completion of the rest of the work, I'm going to let you do yourself. Couple of weeks that there are certain activities that you don't need that hand holding, right? Those ones, I will actually record the activity as a video ahead of the time and provide that to you, right? And still make myself available for you to ask questions and stuff like that. But most of the time we are going to meet like this, right? And Blackboard is how we are going to keep as a platform to upload your assignments, to upload all the materials that you need to follow through, right? And then lab activities, lab exercises, right? So some of them I'm going to group you, right? But most often, right, I'm going to actually make you work individually, right? Because that's actually convenient, especially since we are not in the same room, right? So the practicals are going to be also provided through Blackboard, and we will cover the format and all of that, down the road. And chances are the exam or the practicals are going to be open book, right? And you will have plenty of time to study and plenty of time to complete that as well. And again, just to prevent any overlap with your other activities, we will use the designated lab hours, a designated lab day for your practical, right? So instead of normally meeting on those practical days, you will take the instead of normally meeting during those lab hours, you will take the practical. So that kind of helps you to avoid any time conflicts. And I will come back to the notebook part, but just to highlight the key points, right? So you are required to keep a lab notebook, right? So I'm going to be a little bit more flexible with regard to how you are keeping the lab notebook. Earlier, in previous semesters, I actually make you buy those like actual commercially available lab note books, right? But and then I prefer that you handwrite, take notes, like draw your drawings in hand, and to draw diagrams in hand, and to physically write down notes. And that option is still available, right? But if you prefer keeping that electronically, I'm also open to that, right? Preferably handwritten, because that's actually how you keep lab notebooks, right? But if that is a struggle, but if you prefer, if you personally, it's convenient or a time saver or a lifesaver, if you want to keep that as a Word document, I will accept that as well, right? For example, those of you actually like taking notes on your iPad, like doing hand drawing on your handheld devices, that option is available, because like I do that, like particularly for drawing, I use my tablet to make drawings, right? So I'm just adding that option as a lifesaver, right? It's up to you. But if you start with something, continue the same trend, otherwise you're going to lose track of things, right? If you start electronically, do it electronically. But if you start manually, we do it manually. Don't switch, because then you are going to lose track. Like, okay, where is plant stuff? Where is animal stuff? So keep, adapt one format, continuously follow it.
Can I interrupt real
Yes,
quick?
of course.
I know that last year a professor had like the actual app called Notebook where he created like this and shared the link and we would all go in there. Could we use that? I don't know.
Absolutely, absolutely. Actually, I do not have that link. I need to ask Ron if he has
Yeah,
that.
he was a professor that did it back.
Right,
And
right,
then
right, right.
because it was easier for him to go in and like especially at the end.
I will ask him about that, but if you have like, I mean, that's another thing I wanted to mention. You actually kind of read my mind. If you guys have developed your own formatting of keeping notebooks, that's fine too, right? The Word doc is kind of an example that I used, right? Because you can actually draw hand-free drawings on Word as well. It's more adaptable to different other ways. But Monica, like, do you remember the name of that app?
I think it's just Notebook. I can just search real quick on my computer.
Please do and send a chat. So
It's on Blackboard too. It has like a link at the bottom.
yeah, I will check that
Go in
out. I haven't used that honestly before.
Blackboard tools and then yeah, OneNote class notebook. Yeah.
Okay, very
Like
cool.
this purple thing and it says OneNote.
Yeah. Oh, OneNote. Yes.
Yeah.
I know OneNote is a freaking fantastic tool. Yes, I use it quite a lot. It's freely available for all students in BSU, and I use it a lot for note keeping to keep track of things. Actually, it's a heavily underused tool. Thank you for bringing that up. Actually, completely slipped my mind. Yes, it's a great tool. Freehand. Like it allows so many things, and you can actually keep different tabs for different lab activities. Literally can be maintained as a notebook, right? Yes, that option is definitely available. Definitely available, but yes, of course. And then in terms of, if any of you are wondering, like, I mean, Monica might justify what I'm about to say. Oh, is it hard to use? Do you require additional training? If you can use Word, using OneNote is 100 times easier. Right?
It definitely is, especially with the tabs. And you can add like inside the tabs, you have semi tabs. Like you can like if the handout has like questions, plus you have that additional question. It's much easier.
Yeah, and online, if you search like tutorials to how to use it effectively, you will
Yeah.
find so many, like YouTube videos, so many. And you can, like, it's not like in Words where you type a particular paragraph, it stays there. No, you can drag and drop it around, right? Anything you draw, you can drag and drop it around. Man, it's such a terrific tool. So like for freehand drawing, yeah. Yeah, good job, Monica. That's actually, I don't know why I did not think of that. Yeah, totally available option. Any other questions that you wanted to bring up right now? All right, cool. Let's move on. So yeah, if you actually have a question, so I'm reading in one screen and you guys are on another screen, so I might not see you if you raise your hand. So like voice, interrupt with your voice. So
I have one more question.
yeah, shoot.
I think it said somewhere that it had to be a composition notebook, but does it have?
Good question. You're right. It does say a compositional notebook. That's if you make a physical notebook, right? We recommend compositional style. Compositional styles are like, like kind of in terms of size, it's the same size as your lab manual, like copy paper size. It has like the spiral bind. Now, am I going to penalize for someone using something that is not compositional style? For those who are going old school for keeping a physical notebook? Absolutely not. Absolutely not, right? If you keep any physical book for the lab notebook, no, I do not care what exact generic name that is. Not at all, right? Not at all. Make sure it is large enough to do the writing, to do the drawing, right? Any smaller, like pocket-sized notebooks, that's not recommended because it's way too small, right? Does that answer your question, Emily?
Yeah.
Yeah, alrighty. So those who actually maintain lab notebooks physically, right? How do you submit it? If you create a Word doc or a OneNote, you can submit that as a file, right? A single file, you can upload it. You don't have to convert it to anything. But if you keep a physical notebook, handwritten, one, if you have access to an actual scanner, you can scan all the pages and upload it as a PDF to Blackboard, right? But most of you may not have access to a scanner. The other option is use your smartphones or your tablets and then take individual photographs of every page and then use some of those free tools I have listed in the syllabus to merge different pages, right? Now that's not hard to do that. It's just time consuming and painful to do it. But if you wanted to, but if you wanted to keep, like handwritten notes, you can use that avenue as well. So what I'm trying to do over here is actually provide multiple opportunities, right, for you to keep a notebook. And notebooks are due when? Before there are due dates. Due dates are mentioned actually in the syllabus as well as in the submission pages, right? The due time for everything except the exam is 11:59 p.m. I haven't decided the due time for the exam yet. I'm trying to make the due time the actual lab time frame, but everything else 11:59 p.m. Now that's beneficial. For example, the homework that you are going to start today is due next Tuesday, next Thursday, 11:59 p.m. So if you have questions, even on the 11th hour, right, you can actually ask that questions during this next week's meeting, right? So that you actually have that advantage, that benefit, right? Since you have an extra time, it's not due on the day, it's not due when we meet, right? No, it's actually during the 11th hour. So I'm providing you more time to ask questions from me. Alrighty. So attendance, it's, as you can see, it's extremely important that you not miss, right? It's really important that you are here, right? So class cancellation. Now, the reason this is coming up is, sometimes, I mean, how many of you in your hometowns lost electricity with a snowstorm? Right? So if that happens, we cannot meet synchronously, right? So in that case, I have to actually either, like, we have to decide between myself and Ron whether we are canceling that lab or providing alternative ways to complete it asynchronously. So that's a decision we will make based on what the school decides in terms of the course conduct, right? Sometimes the school cancels just in-person meetings. In that case, we are going to meet. And in that case, I'm also going to record the presentation, right? So that those who cannot make it have access to that. And if the school decides no virtual, synchronous or asynchronous, whatnot, right? Then we'll cancel it. So I'm going to essentially follow whatever the school decides. How to catch up? I will inform you that after I talk with my friend Ron, okay? So regarding special needs, right, if you are one of the special needs students, do read that particular part and contact those persons listed. And if you carry an accommodation letter already, send it to me, right? Chances are we are going to be all set even if you carry accommodation letters, because you're going to take open book exams, you are going to work on your own, you are not going to be physically in a room, right? Nonetheless, send me your accommodation letter, then we will discuss about how to accommodate you personally, right, in private, right, because it's a private conversation. So yes, we do actually apply the plagiarism regulation, right, and do read about that penalization process. So this is the lab schedule. Like I mentioned, unless we are hit with a snowstorm and like cancellation of virtual classes, we will stick to this schedule. Now the schedule tells you when the practicals are going to happen, when the lab notebooks are due, when homework is due, right, and everything else, right? As you can see, the practicals are scheduled within as a part of your regular lab times. And the very last day is actually not a real lab. It's when you present your project presentations, right? Now, again, we haven't gone into detail about the project presentation. Note that all students are supposed to be present for poster presentations, and that poster presentation will happen virtually, right? Where I will be not, presenting anything. You guys will be presenting things in group. Okay. Oh, we actually have someone joining very late. So, alrighty. Any questions? So I quickly went over the syllabus. All good? Alrighty. So next part is actually going into a little bit more detail about the lab, the keeping the lab notebook. That's quite important because it starts literally from today onwards. Okay. Now, again, remember that you have, the option of keeping virtual, like electronic lab notebooks, and you have the option of keeping a physical lab notebook, right? So what I'm explaining applies either way. And honestly, electronic lab notebooks are easy to keep, easy to modify, easy to maintain, unless you learn better as a physical copy, right? I have seen students. My observation, since I earlier only did this like physically, I feel like, it's a better way to learn, but maintenance management electronically, it's convenient. Just my 2 cents for you. So you will use this lab notebook to keep most of your lab notes, right? Pretty much everything you write down can go in there. And you are more than welcome to use that lab notebook during your practical exams. I said that's open book, right? And even when we were teaching in person, we permit you to bring the lab notebook, right? Nothing else but the lab notebook, right? And we apply the same for these online practicals as well. How do you set it up? First, you number your pages, right? Right from 1 to like in wherever it ends. So keep numbering your pages. So the very first pages in your lab notebook are going to be dedicated to table of content, right? Leave about, 2 sheets, right? It's okay to go a little bit more, at least 2 sheets, right? 2, 3, or like 4 sides, 5, no more than I would say 6 sides, right? 2 to 3 pages at most. And then, and it's a table of content. So what you do is actually you mention the first lab, evolutionary trees, right? And then you can even go into subtopics, right? What you did under that particular activity, and then have like this particular activity or this particular exercise in this page, this exercise on that page, like that. So content of pages is something you will keep updating, right, as you complete different labs. And then practical study notes, right? Now that's an optional part, right? The content, content of table, lab notes. What do you take as lab notes, right? So, so the introductory lab is evolutionary trees, right? And then you are going to follow up with not plant labs, animal labs, and then with plant labs, right? So things go in there includes the drawings you make for each exercise, right? The question, when you read the lab manual, it actually asks different questions, right? When you come for the next lab, you are going to read the lab, the handouts, right? And that handout contains certain questions, right? Certain pre-lab activities that simple is to do activities. All of those can go into the lab notebook, right? And during the lab, any drawings you make, right, can go in there. Any labeling you make, right, can go in there. And then practical study notes, and it says optional, right? So anything that actually I explained to you, right, the introductory lectures I'm going to give, right, any additional information I provide, right, anything outside the handout activities, exercises can go in there, right? So it's optional. So if you have that part, I will grade it. I will actually consider awarding you additional points for that, but it's not required. Now, terminology that's required, meaning any new term that you learn will go into the terminology section. Any new scientific term, right? like phylogenetics. Well, I don't know what that is. So you can actually provide, like maintain it as a like a table, maybe like one part, the term, the other column, the definition, something like that. And you might want to keep like, we will learn a lot of terms. So you might want to allocate about like, at least 5 pages, meaning 10 sides, right? To that. And the last part is the species list, right? Where you actually contain information about any new species you learn, right? And you will be coming up with so many species. So any species that you have there, you can actually include information about that species under that species list, right? So I'm not going to read this out to you because this is something you can read on your own, right? So this is actually should be the very first few pages in your lab manual. If you don't have the lab manual with you, right? In your Blackboard page, if you go to course contents, right? Go to lab manual right over here. This contains those pages, right? So just a couple of more, like, and I want to mention is regarding species list, right? So here it actually provides you more detail or more guidance about how to maintain it, right? And some example sketches, as you can see over here. Species list, should contain this particular format. This is the why we recommend this particular format is actually it helps you to study very well, right? The common name of the species followed by the scientific name, right? And then the next column is going to be classification, like what kingdom, what phylum, what class, what order, what family this particular species belong to, right? And then the last column is any additional notes that explains the characteristic of that particular species can go into notes. Anything that you find yourself about that particular species can go in there, right? And that is really helpful when you actually try to address homework assignments, right? So so since we actually have this particular section called practical study notes, the optional part, right? I recommend like anything that doesn't belong anywhere, put in that practical study notes. Don't keep a secondary like like a notebook, because then again it's kind of confusing. Like like where do I put this, right? So anything I explain should go into my recommendation should go into that third part, practical study notes, right? Especially the intro lecture. Any questions so far regarding maintaining the lab notebook? Alrighty, seems like everybody is all set. We did the Blackboard layout, we went over the syllabus, we went over this particular part. Let's now go into...
I'm just a little bit confused.
Yeah, ask the question, please.
About, so is this, the species list is like at near the very end of the book?
So in your lab notebook, okay, species list is the last thing you're going to have, right? It's the last section for your lab notebook. Does that make sense?
So you should like set aside big sections in your notebook for each area?
That is correct. So that's why I actually, if you look at the, like what I'm sharing, it actually, we provide some guidance, like how many pages to leave empty before you designate your sections, right? Like I mentioned, like 2 to 3, 4 contents, right? For lab notebooks, again, the notebook part are another multiple pages, right? About like at least half of the lab notebook. For the half of the pages in the lab notebook should be allocated to lab notes, right? And then practical study guides, well, it depends on how much notes you take. And for the terminology, about 5, 6 pages. Or 10 sides or 10, 12 sides. Species list is the last part, right? You can actually have as many as you want. Now, here's some guidance. Since practical study notes is optional, since that is optional, you can shift practical study notes as to the last section, right? And allocate about 10 to 15 sheets for the species list, and have more space for the practical study notes, right? I suggest that because practical study notes is optional. Does that answer your question, Emily?
I think so.
You need to, if you don't, you have to tell me. Like, what is the question mark in your face?
It's the lab notes itself. There's gonna be like a different section for each lab.
That is correct. So that is correct. Yeah, yeah, yeah. So like, for example, are you wondering, like, for the intro lab, it might be just 5 pages. And then for the, first animal lab, it could be 10 pages. Is that what you're asking yourself?
Yeah.
Okay, yeah, that's correct. So that's what I said. At least half of the pages in your lab notebook, right? If you are physically maintaining one, should be allocated to lab notes. Let's say you have 100 pages in your physical book, right? 50 of them, right, should go into allocate about at least 50 pages, right, to practical study notes. That's what I'm saying. Sorry, allocate 50 of them for lab notes. My bad. Make sense?
Yeah.
Yeah, right. So if you are maintaining it electronically, that doesn't matter at all, does it? Right? Because you can always keep inserting pages as you go, right? If it is electronically, pre-setting, pre-allocating space is not a problem. Now, Emily, if you're wondering, are you going to run out of pages? I haven't had any student struggling with that. No, I never remember student having to like insert additional pages. No, if you allocated about half of your notebook, or more than half, I'm saying at least half, right? Um, of your physical notebook for lab notes section, you should be fine, right? You should be totally fine. But if you're maintaining things electronically, yeah, this is not a problem at all, right? You don't have to worry about like, what if, right? Are we good, Emily? Or still something troubling you?
No, I'm good.
Yeah, sure?
Yeah.
Yeah, okay. Again, like if you want to like follow up, please do. Let's move on to anyone else, guys? Alrighty, let's move on. And guys, like we just had someone else joining quite in the last minute, and I understand it's the very first day, so there's a lot going on. But I want you guys to be on time, right? And that was not a problem. Everybody was on time, actually ahead of time. So, but I wanted to emphasize it's really important because it's harder to go back and forth. Alrighty, let me just go back. Right. I will open this in slide presentation view and start sharing now. Right, share. Okay, now right, so now we are going to go over today's lab activity, right? So we have already provided you with a handout for that. But before going into specific points in the handout, let's go over this, right? So these are the general activities we are going to cover. These are the learning objectives we are going to satisfy by the end of this particular assignment. So organisms are assigned into these particular groups, okay? This is actually the hierarchical organization of living things. That's the simplest way to put it, right? At the largest, greatest level, you start with domain, right? And there are only, how many domains, guys, are there in the living world? Just 3, correct. Can someone tell me what 3 those are?
Multi-cell creatures.
Yeah, actually, partly you are correct, but actual domains, this is something you will learn in the lecture, right? Archaea, eukarya, and bacteria, right? Those are the 3 domains. And actually, I will we will cover that part in the lecture, or someone other professors will. But these are the three major breakups that we are going to go through. So there are 3 domains, and each domain has multiple kingdoms, right? And animals, for example, belong to kingdom Animalia, right? And then you have phylum, right? Or in plant world, we use the word division. So there are multiple phyla in a single kingdom. There are then classes, multiple classes in a single phylum. Orders, multiple orders in a single class. Family, multiple families in a given order. Genus, multiple genera in a given family. Species, multiple species in a given genus. Okay. Now, is that the end? Are there any grouping below species? Of course there are. I'm not going to go over them. Subspecies, right, are there, right? And then varieties are there. Or are there anything between classes and order? Yeah, of course there are subclasses. And hell, there are sometimes subphylums. There are subfamilies. So these divisions can be further fine-tuned. Absolutely you can. But I am not going to go over that part, right? I don't do that even in the lecture, not over here, right? But I want you to know it is possible. Can anyone tell me an easy way to remember this? Because the order of things is important, right? It's extremely important. Any mnemonics that you can come up with to remember this? All right, I'll give you one, right? You're going to laugh at the very end, right? Dear King Philip came over for good sex, right? So that's actually the mnemonics, right? That I used to remember. It's really important. Why I'm giving you a mnemonics? Because it's actually important to remember the order, right? Kingdom, domain, phylum, class, order, family, genus, species. So again, I'm showing the same thing in terms of an example, right? So domain. If you're looking at Canis lupus, the gray wolf, right, one of the iconic North American animals, a North American iconic wildlife, one of my favorite animals, their domain is Eukarya, right? Now, and then, and Emily, just answering your question, addressing back a point you mentioned for my question, you said like in terms of domain, you said multicellular animals. Well, Eukarya contains multicellular animals, but Eukarya domain contains both multicellular and unicellular animals. Okay. And then kingdom is Animalia because the wolf is an animal. Their phylum is Chordata, the same phylum that you and I belong to. Class is Mammalia because they are mammals. Order Carnivora, the group of animals that eat meat. Family Canidae, the dog family. Genus Canis, the same genus that the dog belongs to, the fox belongs to. The species Canis lupus, right? And well, of course, just to highlight, to iterate something I just mentioned, there are multiple genera within the dog family, right? And actually I'm mentioning, I'm outlining several of those, right? Examples over here, right? So, and again, please don't memorize this. This is an example, right? Regarding how complicated the structure within a family could look like, right? Alrighty. Again, if any question pops up, just interrupt me as I go. Now, we talked about a species. It's really important to understand species concept. It's a really intricate, complicated concept, but I'm going to keep it extremely simple because this is a very basic level class. Biological species concept is actually the one we are mostly familiar with. It's actually based on ability for two individuals to mate and produce offsprings that themselves are fertile, right? So if two individuals, right, of animals or plants or two individual organisms can come together, mate, and sire fertile offsprings, right? Those two individuals belong to the same species. That's as simple as what biological species concept is, right? And then ecological species concept is actually less used in the scientific world. it's an instant where you actually have species that live in the same environment, share similar ecological needs, occur in the similar habitats, and you group them into a single species. That's extremely subjective because species that occur in completely two different environments can actually interbreed. Okay, I'll give you an example. Since we talked about gray wolves, right? There's a wolf in Europe, right? Eurasian wolf, and we have North American gray wolves, right? Are those two different species? They are not. They are the same species, right? And if you actually put them together, will they breed? Yeah, very easily, right? There are two subspecies, but they are the same species biologically, so they will breed. That's why ecological species concept is highly questionable. And again, just because two species occur in the same environment, does that mean they can necessarily interbreed? Absolutely not. It does not, right? The problem with biological species concept is actually one, there are many plants that belongs to completely two different species, have very different genetic profiles, but they can actually interbreed naturally. They can successfully interbreed, right? And the same happens in animal kingdom. Particularly, I'm someone who studies a lot of amphibians and reptiles. There are so many different amphibian species that actually naturally interbreed, right? Anyone heard about the term tiger lion or liger? Yeah, tigers and lions can interbreed, right? Mostly in zoos. People have attempted and they have succeeded, right? Whether that happens in the nature is highly iffy because naturally no longer those two populations overlap. So in zoos it has been done. So completely two different biological species, right, can interbreed and produce hybrids. Okay. Phylogenetic species concept is actually becoming increasingly popular than what it used to be. Those are actually, if two individuals, right, organisms share very similar ancestry, right, if they share genetic similarities more than genetic differences, you can group them as a single species, right? The problem why it is questionable is actually because, like, how do you define the threshold, right? Like in terms of genetics, is it 90% similarity of nitrogenous bases? Well, what about 95%? Why not 99%? Why not 80%? Right? That threshold of similarity versus difference is extremely questionable. I experience that question on a regular basis because some of my research involved describing new species that is new to science. And the best tool we use is actually genetics, right? Molecular sequencing. So we actually have standards among scientists for this particular genus, for this particular taxon. This is actually the threshold, right? I mean, I can go into examples later on, and that also depends on not just the taxon or the genus that you are studying, right? It actually depends on the gene that you are trying to map, right? Is it RNA? Or is it DNA? If it is DNA, is it is it mitochondrial DNA? Is it nuclear DNA? So like that, that kind of threshold is determined on a very specific case-by-case basis, right? It's extremely interesting to know more about that, but for this level, I'm going to check myself at that level. Morphological species, individuals that look similar are grouped as a species. Problem, well, there are many species that are morphologically identical, but genetically they are not even freaking close. They cannot even interbreed. Like if you put, like, there are many salamanders, particularly in the southern Appalachian environment where actually I have lots of experiences. They look very similar, extremely similar morphologically, right? Color patterns, quite similar. Body size, very similar. Habitats, exactly the same habitat, right? But they don't interbreed, period. Okay. And there's a flip side. Sometimes there are two individuals of the same species, like males and females, look very different. There are so many butterflies, insects, where males and females are so much different, particularly in terms of size, in terms of color patterns, right? But they are the same species, they breed. Hell, they are males and females, they are girls and boys, right? Not two different species. So morphological species concept have a lot of subjectivity. Biological species concept is extremely popular. That's what we are going to use for the most of the time. But I do want you to at least appreciate the phylogenetic species concept because it's becoming increasingly popular. All right, let's move on. Now, the biggest chunk of one of the most important aim, or the time that we are going to spend mostly on, is understanding the evolutionary tree. That's what we are looking at right over here. So evolutionary tree, phylogenetic tree, cladograms. Those are interchangeable terms. Now, cladograms, if I were to be anal about this, cladograms is one type of evolutionary tree. It's one type of phylogenetic tree, right? Phylogenetic trees are one type of evolutionary trees, but that's extremely anal. That is a little bit more, way too detailed. But for our purposes, it's completely acceptable to use these three terms interchangeably: cladograms, evolutionary trees, phylogenetic trees. Again, for those who are as anal as I am, the phylogenetic trees are one way to show evolutionary relationships, right? Cladograms are one way to show phylogenetic relationships. Okay, I'll leave it at that. Now, generalize all three terms. What does evolutionary tree / phylogenetic tree demonstrate? It shows the relationship between ancestors and their descendants, right? So in the phylogenetic tree, let's try to read it first, right? On this end, so when I say this, I'm looking at, actually, I'm moving my mouse pointer, my laser pointer. So in this tree, that is actually the basal most node. That basal node that you see over here, number 4, that is the most ancestral, that is the furthest ancestor that you can see, right? And then what happens? Well, it broke into 2 branches. So these lines, right, are actually branches, right? And one branch ended up in species E, right? And the other branch, what happened to that? It actually led into another ancestor, ancestor 3. And ancestor 3 evolved into 2 more subsequent ancestors, ancestor 1 and ancestor 2. And ancestor 1 broke into species A and species B. Ancestor 2 evolved into species C and species D, right? So now I'm going to ask questions just so that you understand. The most recent common ancestor to species A and B is what?
One.
Does everyone agree? Emily says the most recent common ancestor to A and B is 1. Any disagreements? All right, yeah, that's correct. Yeah, how do you know? Well, A over here, B over here. Follow those 2 branches. Follow those 2 branches. Where do they meet? Right over there, right? And then the C and D, they are common recent ancestor. Common recent ancestor 2, right? For all species A through D, what is the common recent ancestor? For species A, B, C, D, for all 4 of them, what is the recent most common ancestor? Hey guys, I need you guys to engage.
3.
Is it 4?
Okay, there are 2 questions. Someone says 4, someone says 3. Question is for A, B, C, D, right? Just for A, B, C, D, right? What is the most common recent ancestor? Again, do what I did. You follow the lines. D, C, A, B, right? And then, right, 1 is only for A and B, 2 is only for C and D. So what is common to all 4 of them? 3, right? Why not D? Because actually D comes after, right? D comes afterwards. So no, if it is A, B, C, D, and E, yeah, 4 is the answer. But if I'm just looking at the most recent common ancestor to A through D, that is 3. Listen to the question, common recent, right? 4 is not recent. If I just said common ancestor to A through D, that's a bad question. That's a very unclear question. Yes, then I should accept both 3 as well as 4 as the answer. When I say recent, Try to pick the one that is quite literally the most recent. Okay. Now, how do you know what is recent, right? So left-hand side, right-hand side, right? Whatever closer to the right-hand side is the recent one, right? As you move towards your left-hand side, right, you are looking at more distal ancestors. Okay. Gerard, you were raising your hand.
Yeah, I just had a question. You said that the farthest back node that you could identify was the basal node?
Correct. Yeah. The further you go, the furthest point in your cladogram is the basal most node. Correct.
Got it. Thank you.
Correct. 100% correct. Yeah. Any other questions so far? This is extremely important. This is very easy to understand, but if you don't get it correctly, kind of, become a problem. All right. So the characteristic that is furthest back in the tree is very ancestral. So this particular character is the most ancestral character, right? And whatever a character that you see in species A, or just in species B, just in species D, just in species C, are going to be the most derived characters, right? The further away the character, the more ancestral that is, right? Further to the right, the character occurs or evolves, the more derived or more recent that character evolved. Derived is more recently evolved, ancestral is evolved a long time ago, right? So another fun fact, certain characters, okay, I'm going to ask this as a question. A particular character that evolves in an ancestor, right? Let's say before evolution of ancestor 4, could it be observable in species A, B, C, D, and E, right? Or would you expect it to be only present in some of those species? Or would you expect this ancestral character to be present in any of these species? What do you think?
Unless there was a point for any of the species to evolve to get rid of that trait, then it should still stay with all the species.
That's an excellent answer. That's an excellent answer. Yes, ancestral characteristics, could actually sometimes certain species retain that ancestral feature, and it is observable in the ancestral feature. Some ancestral features are only observable during their embryonic stages, early developmental stages, right? And some ancestral features actually are lost as they evolve, right? For again, like the more the gap between the ancestry and the current position of that particular species, the greater the chances for disappearance of particular ancestral traits. Okay. Alrighty, let's move on. Now, to teach you a few more terms, one more thing over here, right? So these nodes corresponds to ancestors. All the nodes corresponds to ancestors. The terminals, right? The branch tips almost always correspond to a species that is extant, meaning currently living, right? Now, other terms, a clade. Clade is a group of organisms that have evolved from a common ancestor, right? That may not necessarily reveal a particular taxonomic grouping, right? Sometimes it does. In certain cases, it might not. The blue box I have circled represents a clade, right? Which actually comprises species A through D. Now, does that necessarily mean those 4 species belong to the same genus? Maybe, maybe not. Does it necessarily mean that all those 4 species belong to the same order or family? Maybe, maybe not. Don't try to necessarily believe that a single clade always corresponds to a particular taxonomic unit, right? Lineage is kind of the evolutionary pathway a particular organism evolved or a particular taxon evolved, right? So in my cladogram, the yellow lines, right, corresponds to a particular lineage, the evolutionary pathway species A and B took, right, from its ancestral point. So when you trace the lineage, you can understand what characters are ancestral, what characters are more derived, how many different ancestors did they pass through, right? How many evolutionary steps have they undergone, right? So all of those informations are revealed when you go back in your lineage, right? Are we good so far, guys? Right? Not that difficult, is it, guys? Right. So, now again, having seen an evolutionary tree, let me go back just a second and ask you the question: what is an evolutionary tree? How would you define it? With a diagrammatic representation that shows evolutionary relationships. It's a diagrammatic representation, right? That shows ancestor to descendant relationships, right, among different organisms. And how do you construct it? Based on what? Based on a set of known, well-studied characteristics. Now, I am not going to ask you to memorize the definition, rather understand what I explained in terms of the evolutionary tree. Like when you understand it properly, when you get the logic behind it, memorization of a definition is unnecessary. Put the logic back together, you have the definition, right? What evolutionary tree is a tree, it's a diagram. So diagram part should come to your mind. What the hell does it show? The evolutionary connections, evolutionary relationships. Okay, of what? Between ancestors and descendants. See, when you have the logic, memorization becomes unnecessary. I mean, that's the way I say it, right? So I kind of, as a biology professor, I advocate to minimize memorization and advocate favor of understanding the logic. Understanding the logic could be harder, but it actually pays off down the road. What kind of, okay, I said evolutionary traits are developed based on known, well-studied set of characteristics. Or I would rather say a set of traits. And where do you get these traits from? Many, many different ways. Most commonly, morphological, right? How do they look like? What kind of body parts do they have? What kind of cellular organization do they show, right? How is their relative body sizes, right? Morphology, right? Do they have fins like a fish, or do they have limbs? How many pairs of limbs do they have? How many digits do they have in their fingers? How many toes do they have in their legs? Okay, how many pairs of eyes do they have? Okay, do they have a mouth? Do they have an anus? Do they have both? Morphology. Molecular, right? DNA sequences, right? That's becoming popular, and I'm not going to use that quite a lot. Actually, I'm not going to use that at all as a for the teaching purposes. Chemical, amino acids, sequences in proteins. Look at the proteins we synthesize. Think about the common proteins that is synthesized by us as well as by a yeast, right? Cytochrome B, it's a protein that you used for cellular respiration, right? Can we compare their molecular, can we take the protein out? What is the amino acid sequence in humans? What is the amino acid sequence in yeast? You compare, you are comparing the chemical structures, right? Hemoglobin, right? A protein that is used for carrying oxygen and carbon dioxide. Well, take the subunits of the hemoglobin, unravel the subunit, look at the amino acid sequence, compare the amino acid sequences between different mammals, between mammals and birds, using chemicals to construct phylogenetic tree. So all of these forms can be used, but morphology is what we will be using. You might ask, what about behavior? Yeah, not a bad idea. For example, presence or absence of parental care, right? Do they lay eggs or do they deliver young? That's also behavioral. Okay, do they hibernate? Do they not hibernate? That's behavioral. Are they diurnal or nocturnal when it comes to foraging? Foraging is food sharing, food searching. Nocturnal nighttime, diurnal daytime. Yeah, of course you can use behavior. Not very often do we see that happening, but I don't see anything wrong with that, right? The reason why it is not popular is there are not many divisions you can do, and behavior is extremely flexible. So it's very less conservative. That's why we don't use it very frequently. Is there just one correct tree? Good question. Well, what do you guys think, right? Why don't you answer my question? I say like we tend to take a consensus of multiple trees and look for the best support we can. Yeah, that doesn't answer the question very well. What do you guys think? So seems like there are many different ways you can construct trees. There are many different mathematical principles to construct trees. There are many ways to understand evolutionary relationships, many data sources. So what do you guys think? Is there a one universally correct tree and others are wrong? Or could there be many different correct trees? And tell me, what do you think? Yes, there's only one correct tree. If so, why do you think so?
I think it would depend on how they're classified. If it's either through DNA or through other features.
That's an excellent answer, right? So the answer generally is any different responses you want to share? Any of you guys? Thank you, Emily, for actually participating actively, and Jared too. Anyone else who want to share something?
I guess theoretically there could be a technically correct tree if you just say this is the DNA or this is the evolutionary tree. If there's a theoretic way to have a 100% proof correct tree, but I think that that's probably so complicated to figure out. The best way to get to that is to just get a very large census of what's the best looking evolutionary trees and just get the best or closest to out of the large census, because trying to get the perfect tree would probably be like edging on the impossible.
Yeah, so you are actually, those are very good responses, and you are inching towards the correct answer, which is no, we cannot pick one universally correct tree. No, we cannot say rest of the trees are 100% incorrect. Yeah, some of them could be incorrect, yes, but no, it's mathematically impossible to prove that this particular tree is 100% correct, everything else is bullshit. No, that is not possible, scientifically at least. Could certain trees have more consensus? Yes. Could certain trees have more evidence that are observable? Yes. Could certain trees provide statistical evidence for greater likelihood to be more realistic, to be more evolutionary plausible? Absolutely. That's what we do. Right? That's what we do, because, like, needless to mention, some of evolution has already started to happen. Evolution will continue to happen, right? So unless actually you can trace back and tally up every step of evolution, right? How certain features evolved, how certain genes got mutated, how certain sequences evolved, you can never prove one tree as 100% correct. And in sciences, we don't prove, we rather disprove what is impossible, right? What is unlikely, what is less probable. So yes, you guys actually got the point. And it's important to understand that, because later on you will realize that given a certain set of data, you can still come up with multiple different tree types, right? So again, this is actually a deuterostome phylogeny. What is a deuterostome? I don't know how many of you have gone that far into the lecture, right? The lecture is available, but very unlikely that you have gone that far. That's okay. Deuterostome is a group of organisms, a clade, right, that whose first embryonic opening has become the mouth. Dew is secondary. Stome is opening, right? So opening becoming the mouth becoming secondary opening, deuterostome. So the old deuterostome, the first opening becomes the anus, the second opening becomes the mouth, right? So that's what deuterostome stands for, right? This is actually a phylogenetic tree of a few deuterostomes, right? Again, not for the purpose of memorization, but for the purpose of explanation, right? So you can see that several steps in this evolutionary tree, right? These particular nodes shows certain ancestors, and what kind of ancestral feature or derived feature evolved in those steps, right? So deuterostomy, the fact that the first opening becomes the anus or the second opening becomes the mouth, is an ancestral feature for all deuterostomes. Every deuterostome undergoes that step, shows that characteristics in their evolution, in their embryonic development, right? And some of them have an autocode, right? This is really interesting fact. What happened to that autocode? Well, some of the descendants of this particular ancestor retained their notochord throughout their life. Some of them retained the notochord in vestigial stage. Some of them replaced the notochord with a vertebral column. See what happens to some of the ancestral features. Some descendants retain it. Some descendants modify it. Some descendants completely lose it. Okay. So that's just to highlight, like, and you can actually read this on your own, right? What happens to certain features. And again, endoskeleton, right? It's a highly derived characteristic, right? See how many steps it took before that, right? Deuterostomes, right? And a particular group never evolved that endoskeleton, right? And only this particular group, this particular clade actually, led to evolution of endoskeleton. Again, all the descendants of this ancestor ended up evolving an endoskeleton? No, right? Only this particular clade actually have evolved that particular endoskeleton, right? And did all of them retain it? Yeah, to variable levels. For example, this guy actually has a vestigial form of endoskeleton, right? While this guy actually has a very elaborate endoskeleton, right? I mean, that's actually, again, don't memorize it. I'm explaining what the tree reflects to you. Okay, right. Please don't try to memorize this cladogram. Okay, what kind of characters that we observe when we want to group animals? Out of those all the available characters, what type of characters are most suited? Homology shows shared derived characters, right? It actually, homologous characters can help you to understand how certain characters evolved in the ancestor and what kind of modifications happen to that particular ancestor as new descendants evolve from that common ancestor, right? So homologous characters that are shared derived features, right? Shared derived traits actually help us to reconstruct the evolutionary history. Analogous. Analogous traits or characteristics are the structures that look similar, but their evolutionary histories are quite different. Evolutionary origins are quite different. So analogous traits will not help you to reconstruct phylogeny, to reconstruct evolutionary histories, right? So for example, birds and butterflies have wings and they use it for the same purpose, for flight. But they don't have the same evolutionary history, right? They don't even have the same histological history, histological organization, right? So analogous structures cannot be used for constructing evolutionary trees, right? But homologous structures like feather and wings, right, among birds or among birds and dinosaurs, right? Can that be used to reconstruct their phylogeny among birds and reptiles? Absolutely yes. Absolutely yes. Those flying dinosaurs, and as well as the modern day birds, they are closely related. The wing and feathers evolved in exactly the same way among all the birds and dinosaurs, right? Even non-flying dinosaurs actually had feathers, right? How fun is that? Right? So does that make, okay, so there are dinosaurs that can fly and there are some dinosaurs that cannot fly. So those non-flying dinosaurs and the birds' feathers have the same ancestry? Yeah. Yep. Bird of feather, right? So yes, they actually, so the feathers can be traced back, can be used effectively to reconstruct evolutionary history of birds and reptiles, right? It's that how cool that is. Homoplasy, very similar to analogous. It's again, similar characteristics. but they do not have the same evolutionary pathway. They don't have the same ancestry, right? But their characteristics look similar. In fact, it is homoplasy that gives rise to analogy, right? Analogous traits are there due to homoplasy. So like, how the hell does that happen? You have two distally related or not closely related organisms. They occupy the same habitat. They occupy the same environment. They feed in the same way. They forage in the same way. They move in the same way. So evolution happens in similar pathways. They experience the same evolutionary pressures, but similar doesn't mean same, right? So their ancestry is not the same, right? Evolutionary pathways look similar, right? Follow similar pathways, but not the same path, right? So homoplasy does not help us to deconstruct evolution, right? I mean, let me give you an example. Sea turtles have flippers. Whales have flippers. Dolphins have flippers. Walrus, one of my another favorite animal, have flippers. Freaking seals have flippers. Well, does that mean they actually evolved the flipper in the same way? Well, you may argue, oh, prof, of course they did. Why? They all are vertebrates, right? So they are all are tetrapods. So shouldn't they actually, isn't it the limbs that actually produce the flipper? At a very gross level, yeah, they are all vertebrates. So it actually started in the same way. But if you actually, take an X-ray and look at the bone organization, is it going to be similar? Absolutely not, right? What bones you see inside the flipper is going to be quite different. How many bones you see is going to be quite different, right? Among turtles versus whales versus seals versus walrus, you are going to see those differences, right? So there you go. That's actually, so homologous traits, good. Use it for phylogenetic reconstruction. Analogous, don't use it. Homoplasy, don't use it. Alrighty. Moving on. Difference between a character and a character state. A character is a particular physical trait, a particular physical feature, like eye color, skin color, hair color, number of legs, number of eyes, number of antennae, right? The character state is a description, specific description about that particular trait. Okay, for example, eye color. What kind of colors can you expect? Brown, blue, gray, or black, right? Hair color. What kind of different color combinations would you have? Blonde, black, red, right? Legs. How many pair of legs would you expect? None? 2 pairs? 3 pairs? Or 4 pairs like in spiders? Right? So that's the difference between character and a character state, right? Now, those are actually kind of the background, right? So this actually marks the end of the background. And now we are kind of jumping onto going over the lab activity. Any questions regarding the background information we just covered? Good? Excellent, excellent. So the lab work includes, I'm going to interrupt the PowerPoint presentation and switch back and forth to make it more effective. The lab work involves examining a set of a character matrix, right, that has been partially constructed for you. If you look at the lab handout, right, as I explained, you will know what I mean. And that character matrix, which includes different characters, are partially filled out. Or at least I think 2 of them are filled out, and I think we are going to ask you to fill out 2 or 3 more. And then what you are going to do is you are going to examine in order to do that, you are going to examine photographs. Unfortunately, we are not in person. So you are going to look at some of the photographs that I have already uploaded to Blackboard, right? And then look at those pictures and try to come up with characters and character states to fill the character matrix, and then use that character matrix to tackle some of the questions that we ask for your homework activity. I think now what I'm going to do is let me quickly scroll through this before I decide what to go do. Yeah, let's take a quick break and give you some sort of an activity, right? To do, right? So I'm going to let me ask a question from all of you. Yay or nay? Raise your hand, all right? Or put a like a hand up in your icon, right? How many of you have known each other before? Right? Raise your hand if you know each other. No. Okay. So you guys don't recognize each other. That's interesting. This is the first time that happened, right? So what I'm going to do is actually now going to break you into groups, and I'm going to ask you to turn to the lab activity, very first lab activity. That should be in your printed lab handout, this particular handout. If you look at page, it is page 6, right? But before you do page 6, take a really quick look at page 4 and 5 as well. The reason is page 4, right? It shows you an example character matrix that we already constructed for a certain set of animals, right? And then we have provided the actual character and then actual character state that corresponds to each animal, right? And then what we have done is actually based on that, we have already constructed 2 possible evolutionary trees. Say what I just mentioned. I asked the question, can you have more than one evolutionary tree? Yes, you can have more than one evolutionary tree, right? We can argue which one is likely to evolve, which one is unlikely, but that's beside the point. So, so this example is primarily to make you understand what is a character matrix, what you find in a character matrix, what kind of characters can we list as characters, and how do we break that character into character states. So quickly read through page 4 and 5 very first, and then tackle question number 6 or the activity 6 in your lab handout. Now, in the activity 6, Jared, just I'm telling this to you just to avoid confusion, right? This activity is very similar to what we did before, right? So you might actually have previous experience and you might see some very small differences, but it's a long while ago, right? Right? So I'm saying that just so that you're not getting confused or mixed up, or if you're using an older handout, okay? So I'm going to break you into groups randomly. Unless again, one last time, is there a preferred person or a preferred group that you like to define yourself? Good. I already like you guys. And makes the life easy for me. So I'm going to make breakout groups. We have 1, 2, 3, 4, 5, 6, 7, 8, 9, 10. So we will make groups of, we'll make 3 groups, right? And so certain groups will have 3 people, certain groups will have 4. That's fine. And I'm going to let assignments become automatic, right? And if you, and then we can actually make these groups permanent down the road. Ready? So what you do is, what will happen is it will actually automatically ask you to join a particular group. So just pick that particular group and that will break you up into different groups. And once that happens, what you're going to do is in the Blackboard, right? I'll show you this really first. And then actually break up the groups, right? Right. In your Blackboard, if you go to course content page, right? If you go to evolutionary tree information from course content, and then if you go to organisms for evolutionary tree lab, right? Pick either box A or B. I don't care which one you pick, right? If you like A, pick A. B, pick B. And you know what organisms are in A and B, right? In your page 6, it tells you what's in A and B, right? So pick one of them and then start working on the exercise after reading page 4 and 5, right? I assume that you already read 4 and 5 because I asked you to take a look at the handout. If not, just quickly brush through it and get into the activity, and I will jump between your groups, okay? Ready? Understand what you have to do?
Not completely.
Okay, I will explain that one more time, right? So, so what I'm going to ask you to do is actually, first, in your lab handout, right? You know what the handout is, right? Do you want me to show the handout like online?
Sure.
Yeah, of course, absolutely. Please do ask me if something is not clear, right? It's not like I can like do this and you can see it. So in the course content, if you go to the course content, if you go to evolutionary tree information, this is the handout, right? Okay, that's the handout, evolutionary tree handout, right? And there's a PDF version, there's the Word version, right? So in that handout, I want you to first quickly read through page 4 and 5, right? 5 is actually, evolutionary trees. 4 is actually more information. Quickly read it, right? Don't take too much time. And then once you read it, read the activity on page 6, right? Now, where do you find those organisms, right? If you go into click the folder that is named organisms for evolutionary tree lab, that is inside evolutionary tree information, right? There are 2 boxes, A and B, folders, right? Pick either A or B. When you open that up, right? Let me open it for you over here, right? Can you see what's inside? Can you see what these photos?
Yes.
Yeah. So those are the photograph of all the animals, right? So so based on the box you pick, just pick whatever box you need and then follow the exercise in the handout, right? Got it? Understood what to do? Alrighty. I see people nodding. If it is not clear, you have to ask, right? And listen, I do not consider it personally. I don't think that you've been inattentive, right? I consider that as a good thing, actually. I like when you ask questions. I like when you ask me to explain things back. So, so if seems like if it is clear, I'm going to break you into groups. So one more time, read the pages 4 and 5 really quickly, right? And then go into activity or the exercise on page 6 of the handout, right? And in order to complete that, you have to have the Blackboard open. You will have to have the handout open. And the PowerPoint that I showed you will also be helpful, right? That PowerPoint is also in the same folder as evolutionary tree information. Alrighty. Ready? Even if not, here we go. Yeah, now you should be able to join your groups.
Hello.
Yeah, hey. You are in the main session. Alrighty, guys, we are all back. And you all actually have correctly gotten to the first part of the exercise. That's pretty good. That's pretty awesome. So let's actually, unless you have any questions that you want to ask right now, I'm going to go over the examples of the organisms you just looked at, tell you a few things about them so that you can either continue using your characters, right? Or character states, or reframe them when you work on the homework you're on. Any questions before I jump back? Good? All right. Um, and sharing my screen now. All right. Right. Do you see the Noctiluca slide? Give me a nod if you do. All righty, cool. So Noctiluca, right, is actually also known as sea sparkles, right? It's a marine dinoflagellate. Dinoflagellate, marine means it's actually aquatic, right? So, and then, well, oceanic. And then dinoflagellate meaning two flagella, right? And one of them you can actually clearly see, right? Right up here. See this thing that popping out, like the tiny hair-like thing, right? Today I'm going to get the pointer over here. And then commonly occurring bioluminescent and occurs in coastal environments. So where in marine? Coastal largely. Bioluminescence is actually, they can actually kind of reflect lights, right? Like very, can be quite colorful when they are bioluminescent. And then there's actually something called the peristome. This groove is actually called the peristome, right? Inside that peristome is the mouth, also known as cytostome, and the flagella, right? That a longer flagella, right, actually is coming out of it, right? And then other than the flagella, there is also a tentacle, right? So they actually use those two to move around, so they can move around, right? And they use that to capture food, right? And they feed on many different things, you know, bacteria to tiny crustaceans to other algae. And how do they feed? They actually are phagocytotic. They can like encircle other food and then kind of ingest it into their cell. And some of them can actually perform photosynthesis as well, right? That's a really cool characteristics about the group dinoflagellates. Some of them can actually photosynthesize, can eat stuff and can also photosynthesize, right? So what kind of characters can you use over here? Think about that. And I will actually reflect back on all the common characters at the end. I'm going to go one organism at a time explaining certain potential characters that you can, characters and character states you can use, right? And again, stop me if something is not clear. Paramecium, this is something that you guys are very familiar with. So paramecium is actually, again, microscopic. Single cellular organisms, unicellular organisms, free living, meaning they are not parasitic, and they are protozoans, right? They have a nucleus. This guy too has a nucleus. This guy has a nucleus. Their shape is sort of can be like oval shaped is actually the best way to describe them, with rounded or pointed ends, because one end is sort of pointy, other end is sort of roundy, right? And so the shape can be used. They have cilia, so presence absence of cilia, right? And they use cilia to move as well as to feed, right? Eulotrix, it's a filamentous, so that's a shape character, filamentous multicellular green algae, right? Filamentous multicellular green algae occurs in both marine and freshwater environments. So aquatic, but can occur in both marine and freshwater. What you cannot see in my picture or in the slides is actually the fact that they have nucleus, right? They have a vacuole, a central vacuole, which is actually kind of a space where they store stuff. And they have, what you can see in the slide is large, huge aggregations of chloroplasts that perform photosynthesis, right? Claviceps is a very cool thing. It's actually a fungus. It looks like a mushroom. If you thought it looks like a mushroom, you're actually not that wrong. It's not a mushroom per se, but it's like a mushroom because it's a fungus, just like a mushroom actually is, right? And what you're looking at is the fruiting body, reproductive unit of Claviceps, right? The English name for this guy, the common name is ergot. Ergot of rye is actually the common name that I would like to use. And it actually contains toxins. It produces an alkaloid, right, which is toxin. Anyone can tell me a fun story, fun fact about Claviceps that is related to where we live, Massachusetts?
Didn't it make some people go crazy?
It does. It does. What town can we think about a particular town, a historical town in our state that actually relates to something really funny? Anyone heard about the town Salem, Mass? What is the story about Salem, Mass?
The witch hunt.
Yeah, it's real, right? So the chances are people accused, these women as witches who actually, witchcrafted and make people go crazy. It's not the case. The chances are, in fact, the rye they were feeding on actually had this fungus, right? And it's when you actually, if you look at my diagram, the last picture, right? Can you see this, the ear-like thing coming out of the actual corn, right, of the kernel? That is the fungus. That is the fungal fruiting body, right? That is the fungal body. So you don't recognize that as a fungus. You think it's part of the rye, right? So you actually harvest everything, you grind the fungus, you make food out of that, including the fungus. If you put the fungus in, you also have the toxin in it. So when you eat this stuff, it doesn't kill you, it actually impacts your nervous system, right? Like somebody correctly pointed out, it does make you go crazy, right? It completely, you loses your voluntary movements, you show involuntary movements. It has extremely high psychedelic neurological effects, right? So yeah, so essentially you go crazy. There we go. Fun fact. So it actually can stimulate your smooth muscles. So it can impact your breathing activities, your muscle contractions everywhere in your body, right? And so yeah, it's a really cool thing to know. So it's a fungus, so it's multicellular, right? Creates distinct reproductive units called fruiting bodies, right? So those are many different characters that you can actually use to characterize this animal, right? Oscillatoria, blue-green or cyanobacteria, blue-green algae, which I don't like using it, but I put it just for information. The correct term would be cyanobacteria, freshwater species, or aquatic species, but largely freshwater. They also occur in hot water springs, and they don't have a nucleus, right? They do have chloroplasts. Because of that, they can photosynthesize. They have filamentous shapes, but the filament do not make branches. In contrast, if you go back over here, this filamentous green algae can make branches. This filamentous Cyanobacteria cannot make branches, right? And they are unicellular. Don't get misled. They are unicellular, unlike the green algae, which is multicellular. But they make colonies, right? So again, if you go back to this guy, unicellular does not make colonies. Unicellular does not make colonies. Unicellular does make colonies, right? So a lot of information. Euglena, flagellated unicellular organism, does not make permanent colonies, period. Unicellular has a flagella. You cannot see the flagella in this guy, right? But you can see the flagella over here. Can you see the shadow in my picture? In the right side picture, there's a shadow, right? That is actually the flagella. So they can move. When you see flagella, whenever you see cilia, that animal, that organism can move. Freshwater, right? And sometimes you also see them in brackish water or saline environments. The shell shape is elongated. They have a nucleus and have chloroplast, can photosynthesize, right? And the red thing you see over here is actually not the nucleus. It's called eye spot, right? Which is actually has pigments. So if you have chloroplasts, by default, that animal has pigments, right? So if you have pigments, are you looking at an animal with chloroplasts? Not necessarily the case. For example, the pigments in the eye spot cannot photosynthesize, right? Cannot produce chloroplasts. But pigments in the chloroplasts can actually help photosynthesize, can trap light radiation. Diadems, and some of these organisms may not be in your box, right? So don't worry if it comes up. Diadems are algae. Unicellular, makes colonies. Has a cell wall, very unique cell wall, which contains silica, the same stuff you find in sand, right? And they are really pretty. The silica makes different patterns in the cell wall. That's why they actually look really pretty on the sides to the right side. Didinium, right? Again, you might not see in both boxes, only common in some boxes. So they can actually move around. They actually move really fast using cilia. They are protozoans, and they actually feed on paramecium, right? And they have a nucleus, and the nucleus is actually kind of band-shaped. I actually labeled the nucleus in the diagram. Then Laboria, you might actually be, this could have been confusing because it's actually a lichen, right? So it's two organisms, not just one, right? It has a lichen, has an algae in it, and has fungus in it. So that's two organisms. It's a mutualistic association, right? So it can photosynthesize, it's multicellular, it's an organismal association, not just one organism, right? So essentially what you're looking at is two layers of fungus sandwiching the algae. That's kind of very basically the rough organization. So on the right side, you are looking at a cross section of a slide of Laboria. On the left side, the photograph is actually how an actual lichen looks like to the naked eye, right? So lungwort is actually the English name or the common names of that particular lichen, right? Let's summarize every character and character state that we just talked about, right? And then this will actually help you to understand what we talked about today. Can they move actively? Well, if they can move, that's one character. If they cannot move, that's one character state is yes, they can move. No, they are unmovable. Those are the character states, right? And if they can move, then you can ask another follow-up question. Do they have cilia? Right? Yes, no, that's the character state. Well, if they don't have cilia, do they have flagella? Right? And if they have flagella, right, that's actually yes or no, that's the character state, right? And then again, another character is actually could be, are they, what is the cellular organization? Are they unicellular or are they multicellular, right? And then if they are unicellular, you can ask the question, are they colonial or are they solitary, right? Or non-colonial or not, like, or singular. And if they are multicellular, well, of course, then you can ask the question, is it a single organism or is it an association of multiple organisms, right? And then you can ask the question, do they have chloroplasts or pigments, right? That's another question you can ask. And then you can ask the question, like, do they have organelle, like nucleus present, nucleus absent? How about the cell shape? Or how about the organism shape? Better to say organism's shape rather than the cell shape. Are they filamentous? If they are filamentous, are they filamentous branching? If they are filamentous unbranching? If they are non-filamentous, then what is their cell shape, right? Are they elongated, non-elongated? Well, if they are elongated, do they have two pointy ends or do they have actually one pointy end and one round end, right? Like that, you can actually keep creating character states into sub-character states, and continue on. So yeah, those are actually some examples how you can consider some examples that you can consider as you move on to down the road in the homework assignment, right? And we're actually very close to the ending time, very close to 1:45. It takes almost the entire time. So the last slide is actually showing you what the English name, kind of the closer English name for all the organisms, right? And what group they belong to. It's kind of helping you to keep your lab notebook in a better format. If you don't have any questions so far, I'm going to use my last 15 minutes to go over the homework assignments, which also includes creating the evolutionary tree. Any questions? Speaking of which, I don't mind staying after 1:45. I have a meeting at 2, but I can stay extra 15 minutes for you, but I don't want to make you stay with me.
When filling out in a homework assignments, should we use like a different color to indicate that those are the answers we put?
Yeah, I mean, you don't, yeah, like, I mean, most of my students use a different color, like a blue color or a red color. I think the way I programmed it to appear in a different color, but sometimes when you save it on your laptop, it changes. But good question, Emily. Completely okay. And I advise that you use a different color to answer your questions. Anything else? All right. Seems like we don't have much questions. So let's move on to the homework assignment and effectively use the last 15 minutes, meaning the project part I will explain next time when we meet next week. That's my... It's not that hard, right? A little bit challenging, a little bit confusing at the end, but it's actually fun and easy to do. Remember, you always have the opportunity to ask questions from me. Right? Are you looking at the homework in my shared screen? Yes? Give me a nod or a thumbs up. Yeah, thank you, Emily. So here, yeah, you put your name and section, right? And then you have already done this, right? And many groups actually already talk to me that they actually have good character states. If you are comfortable, if you like them, great, go ahead and use the stuff that you already picked. If not, you can change it according to the information I provided, right? Now, evolutionary tree, I know at least one of you groups have already created an evolutionary tree. So what you do is, when you are creating the evolutionary tree, look at all the character states, all 5 of them, right? Jared, go ahead, shoot.
It looks like the slide that you were on changed to wetland looking research thing.
Oh,
it was on the homework page.
Yeah, thank you. Yeah, so it's not the same thing I wanted to show. Sorry.
It was on the homework page though. It just changed all of a sudden.
Weird. Okay. How about now? Now in the homework page?
There we go. Now it's the homework page.
Yeah.
Perfect. Yeah. Thank you, Jared. All right. So use the all 5 character states, right? And again, look only the organisms in your box, right? This actually might contain organisms that are not in your box because we have 2 boxes, right? And then you create the evolutionary tree. These boxes, right? These 5 boxes are for you to name the 5 organisms. And then on the left side, you start drawing your evolutionary tree, right? Again, you can actually, use in your Word version, you can actually like use these lines to connect and draw, or you can even like use the freehand drawing. It's completely up to you how you draw it. How did you do it, Jared? Did you like hand draw it, like when you did it earlier?
I just hand drew it, yeah, and then I'm
Yeah.
going to copy it either into Word or use OneNote and copy it over from one to the other, or something like that.
Yeah. Let me give you a tip when you are creating the evolutionary tree, right? Look at the character states and look for a particular character states that split one organism against the other, right? Or 2 or fewer, or like fewer organisms against a lot more other organisms, right? That's one direction, right? For example, let's say single species or an association. So if you use that character, you separate the lichen from the rest, right? And then and if needed, I can actually at the end I can go through one example for an evolutionary tree. So and then whatever the rest of the organisms left, you can go with are they multicellular or unicellular, right? And then for the unicellular ones, you can decide, come up with a character like, are they colonial, solitary, right? And then you go back to the multicellular ones and then look at something like organism's shape, right? Do they photosynthesize? Do they not? Right? Do they, can they move? Can they not? If they can move, how do they move, right? Do they have cilia or do they have a flagella, right? So like that, you can actually create a particular character to break them into 2 groups, and then each of the branch, you actually break into 2 more groups. Go back to the other branch, break that into 2 groups. Keep breaking that until you end up with the branch tips, where each branch tip has a single species, right? And the example that has worked out in the handout will also help you to do that. And again, if we have time, or even if I run out of time, I will show you an example for one of the boxes and do that with you guys. Okay, so far so good. All right. So then the questions are actually, well, based on your cladogram, which 2 organisms are most closely related? Okay, so how do you know that? Well, if they actually have a much shorter branch depth, what the hell is a branch depth? Let me show you with respect to my PowerPoint slide, right? I'm quickly switching into a different slide, right? Are you looking at the PowerPoint slide, guys? I just want to make sure I'm not showing something else. Are you? Yes. So what is the closest relative of species A? Tell me. What is the closest relative of species A?
That would be species B.
Correct. What is the closest relative of species D?
C.
C, right? C, correct. Correct. So, closely related species coming from a single shared ancestor, right? So, any 2 species that is coming from a recent shared ancestor are closely related species. So, for this particular graph, if I were to ask, what are the 2 organisms that are most closely related? You are going to say A and B, correct? Or C and D, correct. But B and C are not correct. Their common ancestor is actually over there, 3, right? At the point 3, right? Again over here, right? What is the recent common ancestor for Homo sapiens in this particular diagram? Don't feel shy to answer, guys.
Radis, Norm.
Yeah, Rattus norvegicus. That's correct. The Norwegian rat. Correct. So you got the point. And let's move on to which two organisms are most distally related. Again, the further apart, for further the depth between the two branches, between the two branch tips, the further apart they are in the evolutionary relationship. The further the common ancestor to those two branch tips, the less related they are. That's how you know, right? So in the diagram in the PowerPoint slide, what two organisms, I know you are going to butcher the names, I will too. What two organisms are least closely related in the cladogram? Which two branch tips are the most further apart?
Probably either one of the Homo sapiens or anything that's as far into that depth after the notochord, and the Nephetis home
Yeah, it's actually, that's correct, right? The least related branch tips are Homo sapiens and Nephthys, right? The annelid, the worm, right? Those are the least related, right? So within the chordate group, right, the least related are Homo sapiens and Brachiostoma, right? Among Urochordate group, it is Acidaceae and Helocynthia, right? Again, the deeper the differences between the two branch tips, less related they are. Again, if the common ancestor is, way down in the evolutionary tree, the least related those two branch tips are. Okay, let's move on to question number 3 real quick. What traits did you use as derived traits? Does this make biologically sensible? Explain your answer. Which traits did you use as derived traits? Well, remember, like most of the traits that you use are actually derived. Like everything we discussed are derived. They're coming from they have been undergone modifications, right? After their common ancestor, right? So things like flagella that we discussed, that's a derived trait, right? Like undoubtedly a derived trait. Multicellularity, cell organization, derived trait. Organelle, nucleus presence, those are derived trait, right? So yeah, so most of the characters we used are derived traits, because ancestral traits are hard to use, because ancestral traits might be common to most of the species in your box, right? So like what I say in the parentheses, right? It is the derived traits that is extremely useful for you to separate differences in organisms to create the evolutionary tree, right? So that's the answer to the second part. Like explain your answer, there you go, right? Derived traits help you to understand evolutionary relationships better, right? Again, feel free to elaborate things in your own words. What I'm doing right now is not giving you the correct answer, but pushing you towards the correct answer. You just encountered an organism named Hydra, and Hydra are animals. As such, they are multicellular, have multicellular nucleated cells, lack cell walls, and are heterotrophic and stores energy in glycogen molecules. So that's true description about hydras. Using your constructed tree, what lineage would hydra be most closely related to? Explain what trait supports this relationship. So based on your tree, right, not the example trees, your tree that you come up with, where would you put Hydra, right? What particular lineage, right? You can actually describe the lineage by kind of highlighting or painting that lineage in a different color. Remember, lineage is the branch pathway, right? What lineage would you include Hydra? So you can mention that you can either quickly redraw that particular phylogenetic tree here and point and color the lineage, right? Or go back to the tree you drawn previously and point towards, a lineage, or circle the lineage, or put a different color around the lineage to which Hydra belongs to. So how do you know? Look back at the character states, right? You can even, right, like I showed in my example, cladograms, you can include where a particular character evolved, right? Right, in the phylogenetic tree. Where did multicellularity came up? Where did um, nucleus evolved. Where did you start seeing mobility like that? Follow where certain traits evolve. How do you match those traits to Hydra? And based on that, what is your best judgment as where to include it? Now, that judgment may not be 100% accurate in scientific information, but think about where your cladogram is and where you would put in the context of the cladogram, right? Don't try to go back to your lectures, find out where exactly Hydra belong and try to fit in there. That would be hard and impossible, right? Think simple, think in the context of your cladogram. Last one, test your evolutionary hypothesis, that is your tree that you constructed. Look up 5 genera species you examined and assign each one to one of these kingdoms, right? So I have provided, like in my PowerPoint presentation, the scientific names of all the species, sometimes just genera, sometimes genera and species. Look them up, right? And then assign them to these groups. So that's something you need to look up, right? You need to Google these scientific names, right? And which kingdom do they belong to? And then does the tree you construct accurately reflect that kingdom level classification? You might not, because your trees are going to be different, right? The reason why it might not is actually could be multiple reasons could be there. Your example species does not actually represent evolutionary closer lineages, right? And the characteristics you use, you might have accidentally picked a particular character that is homoplasy based, not homology based, right? We did not worry whether the character is homologous or homoplasic, right? So you could have an analogous trait over there. That's fine. Don't worry about that. So that's an explanation, right? This particular trait, although we deemed this shared derived, it may not be shared derived, right? It could be it could be a character that evolved independently, repeatedly. So it might not reflect the actual kingdom distribution. So that's actually one example why it is not, right? Or your different examples do not effectively reflect the true diversity of all kingdoms. That's another reason it could be. Okay, that's actually basically it. So please be as detailed as possible in your answer, right? I accept bullet point answers, but I do expect more detailed answers. So we are actually right on the top of the hour, right? 1:45. I'm more than happy to stay behind and explain you. Take one of the boxes and create a phylogenetic tree with you if you wanted to stay behind. So if anyone wants to leave, you can leave. I will give you like few seconds to bid goodbyes. But if not, if you stay behind, I'll explain an example tree with y'all.
Thank you.
All right, James.
Goodbye. Thank you.
Right. Bye-bye. So the rest of the gang,
I guess I'll head out. I need to go shovel the driveway for my folks. So I thank you very much. Have a great day.
Yeah, of course. Bye-bye, Gerard.
Have a good one.
Yeah, you too. Right. Okay, I think I have the rest of the people staying with me. So Emily, why don't you work with me? Oh, Jasmine. Emily, Jasmine, were you in the same group or not?
No.
No. Do you guys see any of your group members like left, or have your group members already bid goodbyes? Anik?
Me and Kenny were in the same
Oh,
group.
great. Yeah, I that's terrific. What box did you use? Because I have two people now.
We used box A.
Box A. Let's go at box A. Now tell me some character states that you actually used for your character matrix, right? What kind of character? So box A, what character states did you use?
We decided to try the colonies, if they're in colony, multicellular or unicellular, and if they are mobile. So if they move via cilia or a flagella.
Okay, those are the three on the top of the two already there, right?
Mm-hmm.
Okay, now all of us work with me. Box A, right, Anik?
Yes.
Yeah. What do you go by, Anik or Annie, or do you have use like a shorter name?
It works. You can, Anik works. Anik
Really?
is what I usually go by.
Okay, I'll call you
You
Anik.
can call
I
me
don't want
Annie.
to like,
You like
give
to
you
use
a
that,
pet
that's fine.
name.
No, if you want to use Annie, that's fine. I have people who call me that too.
Okay, yeah, I might call it because it's easier. But
Yeah.
anyways, so yeah, Anik, so let's actually work on Anik's example. Look at the box A with me on the on Blackboard. Let me share that Blackboard screen with all of you so we all are on the same page. Are we looking at the Blackboard page, guys? Yeah,
Yep.
Anik? Yeah, good, thank you. So based on all these characters, like colonial or not, multicellular or unicellular, mobile or not, and the default characters already given are what, Anik? Kenny? What are the characters that are already given? I cannot hear you. Kenny? What are the characters that are already in your character matrix?
The cell wall and the storage.
The cell wall and the storage, correct. Now, let's discuss this together. Based on all the characters, cell wall, storage, right? Colonial, multicellular, motile. What character, I'm specifically asking Anik and Kenny, what would you first use to split this group A apart?
We used, I think, whether we started with whether they were in colonies or not.
You started with whether they're colonial or not. Yeah, that's actually not a bad starting
I think
point.
we started with that and then we jumped over to unicellular or multicellular because I feel like that would be easier for us to separate them. So.
I agree with you, Anik. So that's what you would use. Like, is that like the starting point you used? Just to understand you correctly, did you use your starting point as colonial versus non or uni versus multi?
We started with colony and then we decided it would be easier to move on and start with instead.
I think that's actually a good idea. I think that's actually a very good idea. Let's start. Like, I mean, sometimes, what happens is you start something, then you realize like you did, oh, I'm stuck. That's fine. That happens in the real life, okay? Like, I mean, when we actually create these trees. So let's start with that. Kenny and Anik, help me out. So we start with unicellular versus multicellular. So what are unicellular? What are multicellular in your box A?
I have paramecium as a unicellular.
Okay.
And then the Eulutrix as multicellular.
Okay. Now, in your box A, you said you have, let's say, what are all unicellular organisms, right? All unicellular, whether they are colonial or not, right? So, Noctiluca is one of them. Like, well, let's look at the diagram. Oscillatoria, tell me, is it unicellular or not?
Which one?
Oscillatoria, is it unicellular?
Oscillatoria. No, we have that as multi.
Yeah, because that's before I explained you what they are, right? Now you know Oscillatoria is unicellular, right? Yeah. So Oscillatoria is going to be unicellular part. And what is the next one? Let's go to down to the next one. Paramecium, unicellular or multicellular?
Uni.
Correct. Let's go to the next one. That is claviceps, unicellular or multicellular?
Multi.
Correct. It's a fungus. It's multicellular. Noctiluca, right? Unicellular or multicellular?
We have it as unicellular.
That is correct. And we only have one more, I think. Eulutrix, unicellular or multicellular?
Eulutrix we have as multi.
And that is correct. Right? So now actually you did this, excellent. You already have your one animal completely separated, right?
Mm-hmm.
Right? So the claviceps, you already separated it apart. Sorry, just going one step back. Eulutrix, you said what?
The Eulutric, it's
multicellular, right? So
yes.
you actually consider Eulytrix as multicellular, which is correct. Sorry, I just had to go step back. So now on the multicellular branch, you have two animals and what? One, two, four in unicellular part, correct?
It's three for unit and two for multi.
Correct, correct. Did I say four?
Yeah.
Yeah, you are right. So three unicellulars, two multicellulars. With me?
Yes.
Very good. Now let's split up the two unicellular, two multicellular ones. How would we separate Eulytrix from claviceps? So Annie and Kenny. Oh wow, Annie and Kenny. Can you tell me based on your character matrix, right? Which character would you use to split those two apart?
I'm sorry. Which two did you say?
Yeah. So right. So we are trying to split up the two multicellular ones, right? We have one lineage with two multicellular animals, that is claviceps and Eulytrix. Claviceps and Eulytrix. How would you separate those two in the species based on your character matrix?
Well, I think we used Eulytrix being in a colony.
That's not a bad. Okay, yeah, that's actually very close. And the other one between claviceps, yeah. So Eulytrix does occur in colonies. That's actually true. First of all, right? It's a multicellular animal living in colonies. It's a very unique example. Multicellular that occurs in colonial forms. So yes, that's not a bad idea to separate those two at all. How about, I'll give you another much more conspicuous one. Does Eulytrix have chloroplast? It does, yeah, right? It's a green algae. It has chloroplast. It has pigments. Does claviceps has chloroplasts? It's a fungus. It does not, right? So here's a better one. Have fruiting bodies, reproductive units, or does not have reproductive units? That's another character. You can separate those two animals. You guys understand what I'm saying?
Yeah.
Yeah, very good. So I will use fruiting body, right? Fruiting body, present and absent, right? Those who have is claviceps, has a fruiting body, does not is Eulytrix. The things you mentioned are not incorrect, right? I'm just picking one just because it's actually, kind of a fun thing to mention and it's easy to observe, right? Now we have 5 animals. We separated 2 already. We have 3 more. How would we, anyone can actually answer this question, but Annie and Kenny might have an easier time. How would you separate Noctiluca, Oscillatoria, and Paramecium?
How they move?
Excellent answer. Excellent answer, right? Which one can move, which one cannot move?
Noctiluca and Paramecium can move and Oscillatoria cannot.
Excellent, brilliant, right? So now your character is mobility present, mobility absent are the character states. For the mobility present, you include Paramecium and Noctiluca, and Oscillatoria goes out as not mobile, right? Now we have only 2 more animals to separate out. That is Paramecium and Noctiluca. How would we separate those two? What character and what character states?
Well, we separated those two first, and we said those ones we used mobility for them because Paramecium has the cilias, and then Noctiluca has the flagella.
Yeah, so don't use mobility, use how, right?
Yeah.
Yeah, so you can actually use you can go 2 ways. One, cilia present. So cilia, right? So cilia present, what? What organism?
Cilia is in the Paramecium.
Correct. And cilia absent, that would be Noctiluca, right?
Yeah.
Noctiluca. Or you can say what? Flagellum. Flagellum present, Noctiluca, flagellum
absent in the
absent.
Paramecium.
You can go for Paramecium. So So, yeah, so those are many different ways. And also you can also go by shape, right? Elongated, not elongated, or elongated, round, or round, not round, right? So those are many different ways to split those two guys. And all your answers are also correct. So again, is this the only way box A can be created? No, right? I mean, how many trees we already discussed? How many characters that we already discussed? So there could be several correct answers. So remember, when you create it, right? No, don't just create the draw the branches, right? And also name the branch tip by the organism, and also name the branch itself or the node itself with respect to the character you used, right? Whether that particular character is present or absent, right? Any questions? So that's actually one easy example. Any questions? Yeah, I had to jump out in a couple of minutes, but feel free to ask a question that I can quickly help you guys out. Emily?
Did you use the cell wall material also to separate Eulothrus and Claviceps?
Brilliant question. Brilliant question. One, do we have to use all the characters in the character matrix? You don't have to. Can you use cell wall material? Absolutely. Because there's a clear difference, right? There's a very clear difference for breviceps and Ultrix, right? What is it? Cellulose and chitin, right? So an excellent question, Emily, and you're 100% correct, right? So don't try to use all 5 character states. 4 of them, fine. 3 of them, fine, right? But you will not be able to use less than that. Anything else I can help any of you guys?
Oh, I think I got it now. I'll make the changes.
Yeah, excellent. Thanks, Kenny and Annie, for working out that example with me. So if you don't have any questions, I'm going to close the session for all of you. What I'm also trying to do is actually upload this recorded session as a YouTube video so that you can actually watch it whenever you want. And do feel free to drop back for office hours or ask me questions if you have any. And also do not forget that you have plenty of time between end of next lab and the homework due time to ask questions. Okay?
Yep.
Are we good at least so far?
Yep.
All right, guys.
Have a good one.
Right. Bye-bye.
Have a good day.
Yeah. All righty. Bye-bye. Good day.